Hello all, welcome. Welcome back to our new video. Today I will be explaining about the apoptosis. Before going to the actual topic, I will be giving a brief orientation about what is apoptosis. Okay, so what is the meaning of the apoptosis? So the when the cells have, this is the cell, when the cell is having a certain problem, the cell is having a certain problem, that is decrease in the cell growth. There is a cell growth, decrease in cell growth is there, but sometimes the virus may attack the cell. Okay, so some problem is there within the cell. So if there is a problem for the cell, it commits suicide. That method of killing by itself is known as apoptosis. Okay, that is the basic definition. So while we are going to the actual definition, the definition says that the process is a programmed cell death in which internal cellular program direct the cells to die in a controlled manner by activating enzymes that degrade the cell's nuclear DNA, nuclear and cytoplasmic protein. So it is a programmed cell death, but it is not dying instantly or immediately, but it is a programmed a series of reactions are occurring. It is a programmed cell death, also it is in a controlled manner. So the definition is that the process in which it is in a controlled manner. Okay, so the process the process is often called programmed cell death, in which its internal cellular proteins or program direct the cells to die in a controlled manner. Okay, in a controlled manner it is dying, not in a instantaneous manner. So we can talk about two types of cell death are there, right? We know that about two types of cell death are there. What is there? One is necrosis. One is necrosis and one is apoptosis. That what we are discussing right now. Apoptosis. So what is the meaning of necrosis? What is the difference between these two? Necrosis means the swelling. Necrosis means the cells will be swollen up. The cells will be swollen up and rupturing occurs. But in apoptosis, the cells will be shrunken and the membrane, the nuclear organelles membrane will be preserved. In other case, it is not like that. Okay. So I will be explaining a diagram is there. So I will be explaining by focusing on this diagram. Okay, so first of all, what is happening? First of all, the cell death may occur due to the external or internal factors. External means not environmental factors. External means this is the cell, right? The external means the factors that are present outside the cell or the internal factors. What are the external factors? So when we are having a cancer, right? So we will be giving radiation. So when we are giving radiation, the tumor cell have to be removed. So in case of radiation, we have to remove the cell. Okay, so the external factors include radiation, or if there is some deficiency or some problem in growth factor, so the cell cannot grow. There is some it is a growth factor means it is an external problem. So if there is a problem in growth factor, and we have to we have to remove the, or we have to die the cell. Okay, so the external factors will remove the cell. Okay, kill the cell. Okay, the internal factors mean DNA damage. If there is any damage to DNA, if there is a decreased oxygen supply, that is hypoxia, or in any stress condition, we have to kill the suicide or kill the that cell. Okay, that are the two factors, intrinsic and extrinsic factors are there. So first of all, we will be talking about the extrinsic factors. What are the extrinsic factors? So sorry, not what are the extrinsic factors, how the extrinsic pathway takes place. Okay, so this is the cell membrane, right? In the cell membrane, we have a receptor is there, a death receptor. The death receptor is THF. THF receptor, the full form of THF is TNF, sorry, not THF, TNF receptor, TNF, tumor necrosis factor. Okay, so in the cell we have tumor. Here you can see a receptor is there. Okay, so receptor is there. The name of the receptor is TNF, the type 1 TNF receptor. So in that TNF receptor, if there is an extrinsic factors, due to the extrinsic factors, the death ligand will come and bind to the TNF receptor, which will initiate the cascades, which will initiate the process of the apoptosis. Okay, is it clear now? Okay, so I told that. mm, I told that, and there is a, so I told that here we can see a type 1 TNF receptor is there, and TNF receptor is there. So if there is any extrinsic factors such as loss of growth factors or radiation, that ligand will get activated, and that ligand will get activated, and that ligand will bind to the receptor, and after binding the receptor, the other processes takes place. What are the other processes? The initial the other we will be calling the other processor which initiate the apoptosis is their caspase processes. The caspase is of two types: initiator caspase. The caspase is of two types: initiator and executional caspase is there. First of all, initiator caspase will activate when the initiator caspase is caspase 8. So after this binding, Casper 8 will get activated, and after Casper 8, Casper 3 will get activated, then apoptosis will be occurring. This is the basic thing that you have to understand. So next, talking about the intrinsic factors. So intrinsic factor, if there is any DNA damage, like that I told, hypoxia, stress. So if there is any death signal, so if there is any death signal, what will happen? We have to, if there is a death signal, we have to kill this cell. So the processes will be occurring to kill that cell. But actually in our body, an apoptotic enzyme is there, BCL2. This is the anti-apoptotic enzyme. So what is the function of this enzyme? This will suppress the this pathway. This will suppress this pathway. So what is the role of this pathway? This pathway is the pathway in which the apoptosis is occurring. So if there is a suppression of this pathway, means the other processes will not occur. Though if there is a suppression of pathway here, means the backward processes will not occur. So so there will be no apoptosis will not be there, cell death will not be there. So we have to remove this masking site first of all, right? This is masking this. So we have to remove this masking site. So for removal of this masking site, first of all, if there is a death signal, this BH3 will inhibit BCL2. BCL2 is the anti-apoptotic enzyme. So if there is a death signal, this will get inhibited, and this process will continue. What is this process? I will say, first of all, BH3 will get activated. Then after activating BH3, BAK or BX get activated. When this get activated, in the mitochondria, the permeability of the mitochondria increases, and the cytochrome C get released from the mitochondria, and after releasing certain apoptotic protease this is a apoptotic protease activating factor will get activated, and the caspase, the initiator caspase and the execution caspase will get activated. The initiator caspase here is caspase 9, and execution caspase here 3 and 7. Okay, then apoptosis occur. So the code to remember the initiator and execution caspase, this is the basic thing that you have to understand. Okay, I will give you a code to remember that. Okay, initiator caspase and execution caspase. Initiator. So actually the caspases are 2, 8, 9, 10, and the execution is 3. First, this is initiation. Execution is this. Execution means 3. So 3 into 6. 6 plus 1, 7. This is the code. 3 into 6 is 6 plus 1, 7. So here first of all it will be starting with the 2 and rest 8, 9, 10. Okay, this is in a consecutive manner. So how we can differentiate between initiation and execution? Initiation, we are studying from the first standard to from lower number is initiation, that you just remember. This is the lower number, number 2. So we can remember like this. Okay, I hope that it is clear for you. This is the basic thing that you have to understand. 70% is over. Then we can read the extrinsic pathway first. The apoptosis is tightly regulated by the process induced by two main pathways. Extrinsic pathway. So first of all, the death receptor initiated pathway, I told that. And then what we can see, mediated by the binding of the ligands with the death receptors present on the cell surface. I have told that most of the death receptors are, and so tumor necrosis factor, type 1 tumor necrosis factor. This is the death receptor which is present on the cell membrane, and the ligand for the ligand, the death ligand which is coming and binding the death receptor is TNF alpha. Okay, I told that TNF alpha. This is the TNF1 receptor for this the ligand is TNF alpha. Then another type of ligand is also there. This is the important thing. This is not that much important. So this is the receptor is fast. Fast L is the ligand. Fast L. Okay, fast ligand is the ligand. Fast is the receptor. Then activation of cascades processes, cysteine dependent aspartate specific proteases. Okay. Then what about intrinsic? I told that there is an anti-apoptotic protein. The intrinsic signals stimulate major anti-apoptotic protein BCL2. That means if a cell is performing in a normal human body, the cells are in a normal state. So if there is no problem, that is, if there is no problem, the anti-apoptotic protein will stimulated. So what will happen? The apoptosis, that means cell death will not occur. That we yes, of course, that have to be occurred. If there is simply apoptosis, means all the cell will get damaged. So to avoid that, BCL2 get activated always. So if the cells are in under stress, if there is any problem for the cell, like that hypoxia, I told no, hypoxia, DNA damage, this, and so after having this type of problems, so this anti-apoptotic protein, which is an inhibitor of apoptosis, will have to be removed, will have to be down regulated. So under the stress and during the stress, this have to be down regulated and replaced by increased production of pro-apoptotic proteins. That means this have to be down regulated, and the factors that increase the apoptosis have to be increased. That is, I told no, BK and BX. Then after the activating, the mitochondrial permeability is altered and the release of cytochrome C and the other process occur. What are the caspases? I told no. Initiators are 2, 8, 9, 10 and effectors or execution caspases. Executase. Okay. Then inflammatory caspases also here they provided. Okay. Then at last caspase 3 is the execution caspase, and it is called YAMA. Okay. This is the thing that you have to understand. Okay, this is just, then what are the biochemical functions that occur in caspases? DNA laddering occurs, then DNA rupture occurs, that we all know that. Okay, this is the difference between apoptosis and necrosis. Necrosis means swelling and plasma membrane rupture. Here membrane is not an apoptosis, it is bulging, shrinking, and there is no plasma membrane rupture. So this is the thing. Then what is the in cancer? So actually the apoptosis will prevent the cancer. How you know? If there is any UV in when we go out, if there is any UV attack in our body, the cells will get damaged. So that damaged cell have to be removed by our body. So the apoptosis will help to remove that damaged body and prevent the cancer. If there is no removal of that substance, the cells will accumulate and it will give rise to tumor. Okay, so in tumor it is really important. So that is the basic orientation, that is the basic thing that you have to understand. Then after understand this concept, this is the basic concept that you have to understand. After understand this concept, we can read this. Okay, normally when there is damage of cell, it is destroyed by the apoptosis. This prevents cancer. When the skins are damaged by UV light, apoptosis is triggered. If apoptosis does not occur, these damaged cells may survive and develop into cancer cells. I have told already, and this is the thing that you have to understand. Okay, the loss of p53 tumor gene leads to inactivation of p53 protein, re-enter the cell, unable to sense DNA damage that initiates apoptosis. What you know? if there is an inactivation of a p53 protein, if there is due to any problem, if there is an inactivation of p53 protein, the cells this lead to unable to sense DNA damage. So what will happen, if there is no if it cannot understand DNA damage, what will happen, and the apoptosis will not occur. Intrinsic pathway of apoptosis will not occur. That's lead to tumor. Okay. The anti-apoptotic, that is BCL2 family members, are upregulated. So what will happen if there is this is the inactivation of this protein. Actually this is leading to apoptosis. If there is an inactivation, this lead to unable to sense the DNA damage, and what will happen, the apoptotic protein anti-apoptotic protein will get upregulated. So this will get upregulated, and it will prevent cytochrome C from releasing, and it will lead to cancer. So this is the basic thing. Then how can we treat cancer? Chemotherapy and radiation forcing cancer cell to undergo apoptosis. If they do chemotherapy, then automatically apoptosis will occur by extrinsic pathway. Then BH3, it is one of the intermediate in the intrinsic pathway. If you activate BH3 protein, it will strengthen the process of apoptosis. Then some drugs that restores pro-apoptotic factors. So I told BH3, if we activate the drugs which activate BH3 will lead to apoptosis, so cancer can be prevented. And also increase the permeability of the drugs that increase the permeability of mitochondria also initiate apoptosis. This is the basic thing that you have to understand about the apoptosis. I hope that it is clear for you. So all the best. Thank you.